So my laboratory studies the function of the DEC or DEK protein. It is a chromatin organizing protein, meaning that it helps the cell figure out how to organize its DNA and how to use its DNA, as well as how to keep it protected and repaired so that it can be used properly. What my work has found in the past is that over 60% of breast cancers, or many, depending on the study, actually overexpresses breast cancer. All subtypes, but most commonly the basal subtype of triple-negative breast cancer, which is the most difficult to treat type of breast cancer. And so we have found that when DEC is overexpressed, we really get a lot of these tumor-promoting activities in cancer cells, that they grow more, they metastasize more, and they tend to be more drug-resistant. And so one of the ways that that happens is that DEC controls the expression of other genes. In particular, we've been studying a pathway called Wnt, or Wnt ligands. And these are signaling molecules that go from cancer cells or other cells and communicate to the cells in the neighboring environment. And they can tell neighboring cells to grow more, to spread, or to react in such a way to promote the tumor growth. And so there are lots of other signals that DEC-expressing cells can send out to the environment that we are now realizing affect the tumor growth and progression in a way that's bad for the patient but good for the tumor, unfortunately. And so the work that I'm presenting today at the San Antonio Breast Cancer Symposium is showing that when cancer cells, breast cancer cells, express this DEK or DEC protein, it's telling the immune cells around it, particularly the macrophages, to change in a way that suppresses the immune system and also supports the growth of the tumor and potentially the growth of blood vessels that would feed the tumor. And so we're also presenting how we think that might happen through multiple signaling factors, and we think our preliminary data is showing that one of those mechanisms may be those Wnt proteins. And it has not been shown before that proteins like Wnt that are typically expressed by triple-negative breast cancers may be partially responsible for shutting the immune system down and permitting tumor growth. When these signals from DEC-expressing cells are sent out into the environment around the cancer cells, they can bind to proteins on the surface of the macrophages. And so macrophages can react to the environment in one of two ways. They can react in a way that's similar to infections, and that creates this kind of cell-killing immune system environment that really kind of attacks things. And that's kind of an anti-tumor approach. And those kinds of macrophages like to sequester or retain iron, supposedly to keep it away from bacteria so that bacteria don't use it for replication. The other way that macrophages can respond to their environment is called an M2 polarization. And when macrophages polarize or react in this way, they start secreting growth factors that help the tissue around it to regenerate or grow again, which obviously would be bad for a tumor, and can also promote the growth of blood vessels to repair that tissue, which also would feed a tumor. These types of macrophages recycle iron, meaning that they take it in from blood cells or other sources and spit it back out, potentially to help the growth and metabolism of the epithelial cells and endothelial cells around it to help repair the tissue. Yes, so we've been working to try to find a way to therapeutically target DEC. It is very difficult because it doesn't have an enzymatic activity that you can block. It's also nuclear, so you can't get an antibody kind of based therapy into the cell to block its function. So the ways that we've been investigating is to use genetic methods to decrease expression of it. And the other possibility is to understand how it works after the fact, all the other pathways it affects, to target those pathways. For instance, Wnt inhibitors. There are many that are in development and that are either in development or clinical trials, and we've have some preliminary data that if you treat cancer cells with those Wnt inhibitors, they no longer express the excess Wnt's when they have DEC, and that prevents the macrophages from kind of reacting in that tumor-promoting way.